Hello, I'm going to speak a little about the basic biology in leprosy. Leprosy is caused by Mycobacterium leprae and it is an acid-fast bacillus. I'll talk about it in very short. The genome size of Mycobacterium leprae is much smaller than its uh, relative Mycobacterium tuberculosis. The number of genes in Mycobacterium leprae has decreased, and this is said to be most probably due to its obligate intercellular parasitic life. As it lives as a parasite, most of the nutrients and most of the metabolites it uses might come from its host, and thus many of the biochemical pathways may have become redundant. Due to that reason, many of the genes have converted into pseudogenes and they do not come into use. The Mycobacterium leprae is usually known as a disease of humans, but it has been found that it is normally found in armadillo whose body temperature is around 33 to 34 Celsius. Even in humans whose core body temperature is 37 Celsius, Mycobacterium leprae is only found in the cooler parts of the body, mostly the ears and the skin and the peripheral knobs, not the central knobs but the peripheral knobs which are cold. The bacteria has also been are also grown in mouse for research purposes. Recently, uh, it has been found that the Mycobacterium leprae is able to grow inside amoeba and more specifically acanthamoeba. Scientists have grown the M. leprae inside pure culture of acanthamoeba, and these bacilli were found to be alive even after 1 month of growing inside the amoeba. In humans, Mycobacterium leprae is more more specifically grows in the macrophages and Schwann cells. The outer surface of Mycobacterium leprae consists of an oligonucleotide linked with the lipids called the phenolic glycolipid. Phenolic glycolipid 1 is very specific to Mycobacterium leprae. This antigen is thought to be responsible to bind on the Schwann cells and also help the macrophage to ingest the bacilli into its phagolysosomes. In the Schwann cells, as shown in the figure, the M. leprae will bind to the outer surface of the Schwann cells and gets phagocytosed into the Schwann cells. The M. leprae, while is said to be uh, infect the nerve cells, but it doesn't affect the nerve cells on its own, but it affects the Schwann cells, which usually piles around the nerve cells. Leprosy is very specifically known to cause loss of sensation or loss of motor function of the human host. But very few scientific evidences has been gathered to know the actual reason why the nerves cease to function. There are many hypotheses, and in one of the article which is referenced here at the bottom describe three main hypotheses that may cause the, the knobs to cease to function. The first one is ischemia. It is said that the inflammation caused by the M. leprae in the swan cells will increase the pressure to the nerve cells which is at the center of the nerves surrounded by the swan cells and thus make the nerves unable to function. The second hypothesis is that the M. leprae causes the apoptosis of the swan cells, as we know that the myelin sheath is contributed by the swan cells and the apoptosis of swan cells cause the nerve functions to cease. The third hypothesis is the demyelination caused by the infection of the M. leprae. It has also been said that the infection of the M. leprae to the swan cells is caused by the a phenomenon where the usually infection by M. leprae causes the cell to turn into stem cells. So the infection of the M. leprae in the swan cell causes the demyelination of the swan cells and thus cause the cessation of function of the nerves. In the macrophages, what usually happens is that when any bacteria is ingested by the macrophages, various reactive oxygen species are generated in the phagolysosomes which kill the bacilli. What are phagolysosomes? Phagolysosomes are intracellular vesicles formed by the fusion of phagosomes and lysosomes. Phagosomes are intracellular vesicles formed by engulfment of extracellular bacilli by the macrophages. This is very similar to those the phagocytosis known to be done by the amoeba. The phagosome inside the cytoplasm of the macrophages fuses with lysosomes. The lysosome contains various hydrolyzing enzymes and reactive oxygen species, and when the phagosome, which contains the bacteria engulfed by the macrophage, fuse with lysosome, phagolysosome is formed, and these phagolysosomes help to kill the bacteria. But in case of Mycobacterium leprae, as shown in the right side of this slide, various processes inhibit the killing of the Mycobacterium leprae bacilli. Among many of the ways by which Mycobacterium leprae prevent itself from being destroyed by the phagolysosomal enzymes is like the bacilli prevent the fusion of phagosome and lysosome. Thus, the phagosome will not have the hydrolytic enzymes and reactive oxygen species, so the bacilli inside them will not be killed. The other is the other known and published studies has found that the bacilli in the phagolysosome prevent the uh, prevent the decrease in pH which is required for the activity of the phagolysosomal hydrolyzing enzymes. In fact, inside the phagolysosome, the pH is low, and this low pH helps the hydrolytic enzymes to work better. But the M. leprae in the phagolysosome prevent the decrease in the pH. Similarly, the bacilli are also known to escape the phagolysosome to the cytoplasm, where the hydrolytic enzymes and the reactive oxygen species cannot work. If the hydrolyzing enzymes and reactive oxygen species would come out of the phagolysosome, then it will destroy the cell or macrophage itself. So the activity of the enzymes and the chemical species inside the phagolysosomes cannot act outside the phagolysosome in the cytoplasm. These and many unknown processes prevent the Mycobacterium leprae to be killed by the macrophages. In the 1990s, uh, it was found that the two types of leprosy, which can be either classified as multibacillary and paucibacillary leprosy according to the World Health Organization classification, and lepromatous leprosy and tuberculous leprosy according to the Ridley-Joplin classification. Uh, these two kinds of leprosy uh, represent two different immunological states in the leprosy patients. Multibacillary patient, which is usually defined as the presence of higher concentration of bacilli in the skin, or as detected by the slit skin smear technique, and also synonymous to the lepromatous leprosy as classified by the Ridley-Joplin classification system, is said to be consisting of type 2 cytokines like IL-4, IL-5, and IL-10. The paucibacillary leprosy, usually defined as having very low concentration of bacilli or lack of bacilli as detected by slit skin smear technique, and also synonymous with the tuberculous leprosy type as classified by the Ridley-Joplin class, have higher concentration of type 1 cytokines, for example IL-2, interferon gamma, lymphotoxin, etc. These two different kinds of immunological state are can be logically related to the two different states or two different types of leprosy classification. For example, type 1 cytokines, which is usually related to inflammatory state of our body, it can be exemplified by very active macrophages. When the macrophages are very active and healthy, they are able to kill or limit the number of Mycobacterium leprae living inside them. When the body is at type 2 cytokine state, there is lack of type 1 cytokines, and the human body has lower inflammatory capacity. Thus, the macrophages inside which the M. leprae are living cannot limit the growth of the bacilli, leprobacilli, and thus the leprobacilli will easily multiply inside the macrophages. Now we'll talk in short about the diagnostics in leprosy. The leprosy is known to be one of the oldest diseases known to mankind, and it is also one of the oldest disease in our scientific history. Just few years before Robert Koch, a German scientist or bacteriologist, gave the germ theory of disease. In 1873, a Norwegian scientist called Armir Hansen had said that he found bacilli in the skin scraping of leprocy patients. Thus, even in the scientific era, leprosy is one of the oldest disease whose etiological agent was found to be caused by or found to be due to some kind of germs. So, in spite of being and having a very good history of scientific evidences, we still don't have the diagnostic kit for leprosy. I'll just try to explain with 3 slides on the uh, diagnostics part of leprosy. Here we can see that the the interferon gamma production by the blood induced with or activated with different kinds of mLepre related antigens. Uh, here we can see that the concentration of interferon gamma as stimulated by PPD, which is the usual M. tuberculosis antigen, or M. leprae sonicate, which is the M. leprae killed by sonication, MLSA minus LAN, which is M. leprae soluble antigen without lipoarabinopamine, and MLCWA, M. leprae cell wall antigen. All of these antigens can stimulate the production of interferon gamma in different patients group, which I'll say what are they. But the concentration of the interferon gamma is too less, very less in lepromatous pole of the lepromatous patient. For example, I will just first describe what are the abbreviations. So EC is endemic control, means healthy people in endemic region where leprosy is found. CT is like contacts. Contacts mean household contacts living together with the leprosy patient in the same family. TTBT means tuberculous and borderline tuberculous patients. BL borderline lepromatous, and LL lepromatous leprosy patients. So here we can see that when antigens are used to stimulate the blood of the leprosy patients, usually for 2 to 5 days, stimulate the antigen is used to stimulate the blood in aseptic condition and at human-like conditions like 5% carbon dioxide atmosphere and 37 Celsius degrees Celsius temperature. The production of interferon gamma by the mm, macrophages is very good in the TTBT or tuberculous patients, but very low or not at all in the lepromatous patients BL and LL. In fact, the production of interferon gamma is also very good at in the endemic controls and contacts. As we know that endemic controls and contacts are not leprosy patients, if the production of interferon gamma is used as a signal for being infected with leprosy, then endemic control people and contact people will be falsely identified as leprosy patient, whereas the BL and LL patients who are very high bacterial load patients will be false negative patients. So the interferon gamma based diagnostics cannot be used for diagnosis of leprosy. These two figures show the concentration of antibodies against specific antigens in different patient groups. Here we see that the concentration of antibodies in MB or multibacillary patients is high compared to the PB patients. The two different sets in the bar graph represent two different kinds of antigens, antibody against two different kinds of antigens. Here we see that the MB patient or lepromatous leprosy patients have very good antibody concentration in the body against the M. leprae antigens, but the PB patients do not have good antibody response, and the concentration of antibody against the M. leprae antigens may be very similar to that of the control patients or TB patients. So, testing the presence of antibody will will correctly diagnose the MB patients or multibacillary patients. The antibody test will give false negative test for PB patients. Very recently, um, Dr. Annie McCulloch has been working for development of lateral flow assay based diagnostic for diagnosis of leprosy. Their group have been working to identify specific cytokines that may be in high or low concentration in leprosy patients, and these expression or high expression or low expression of these cytokines may represent different groups of leprosy. For example, multibacillary leprosy, possibacillary leprosy, or household contacts of leprosy patient. So these antigens, these cytokines are expected to correctly diagnose the leprosy patients, and thus will be very useful to correctly identify leprosy patients and start treatment, and thus will is expected to decrease the transmission chain, and thus decrease the prevalence of leprosy. And this is I think the last slide where uh, I want to show that the M. leprae uh, has been found. Different studies have shown that they are found on the outer surface of epidermis, uh, and the bacilli uh, mm, the transmission of the M. leprae could be seen until 1 month of the start of the treatment of the leprosy patients. The figure in the right is a very good study which shows that M. leprae could be caused by means transmitted uh, through environmental sources. What the study um, is trying to show, the study published in 1998 in International Journal of Leprosy shows that um, uh, in normal children, the sites where children gets bruises or injury when they play, the sites of the injuries, normal injuries and bruises that children get is very significantly correlated to the sites where leprosy lesions are seen in the children of the same age groups. This gives a hint that M. leprae may not only be transmitted through droplet infection, which means from nose to nose or by inhaling the M. leprae from the air exhaled by leprosy bacillus, but the M. leprae bacilli may also enter through the bruises or injuries or cuts in the skin and thus develop at the sites where the lesion develop in the sites where the M. leprae bacilli entered the